Hello everyone and welcome to this second live session of the first International Electronic Conference on Chemical Sensors and Analytical Chemistry. This conference has been organized by MDPI and the MDPI journal Chemosensors, and it is chaired by Professor Nicole Jaffrezic-Renault from the Institute of Analytical Sciences in France. Thank you very much for joining us today. Remember that the conference is now open for discussion. So until July 15, you are invited to check and comment all the papers presented to the conference. I would like to remind everyone that this live session is being recorded and that the recording will be posted in the conference webpage for everyone to watch it as soon as possible. We will have 4 presentations today. And after the presentation, we will hold a Q&A session during which we will address any question or comment submitted by the live online audience. So if you are joining us live, please type your questions in the Q&A box that you will find at the bottom of your screen and at any time during the session. Today's session will be chaired by the conference chair, Professor Nicole Jaffrezic-Renault. Nicole received her engineering degree from the École Nationale Supérieure de Chimie in Paris in 1971 and the doctorate d'etat at science physics from the University of Paris in 1976. She is an emeritus director of the research at the Centre National de la Recherche Scientifique, the past president of the Chemical Microsensor Club, the president of the Analytical Division and member of the Administrative Council of the French Chemical Society. She was decorated as Knight of the Legion of Honor and Officer of the National Order of Merit. Her research activities in the Institute of Analytical Sciences include the conception and design of biochemical sensors and their integration in microsystems. She also coordinates several European and national projects for the development of microsystems for biomedical and environmental monitoring and for food safety. Thank you so much for chairing this conference and this session. The floor is yours, Nicole.
Good morning, everybody. Thank you, Anna, for a nice introduction. It is my pleasure to introduce Professor Kourosh Kalantar-Zadeh, who is the first keynote speaker. He's a professor of chemical engineering in the UNSW and one of the Australian Research Council laureate fellow in 2018. Professor Kalantar-Zadeh is involved in research in the field of material sciences and sensor and has co-authored more than 450 scientific papers and is also a member of the editorial boards of journals including SAS Sensor, SAS Applied Nanomaterials, Advanced Materials Technologies, and SAS Nano. He has received many international awards, including the 27th IEEE Sensor Council Achievement, 2018 SAS Advances in Measurement Science Lectureship Award, and 2020 Robert Boyle Prize of RSC. He also appeared in the Clarivate Analytics Most Highly Cited list since 2018. Please, Professor Kalantar-Zadeh.
All right, thank you very much for the introduction, Nicole. Okay, so today I'm going to talk about ingestible gas sensing capsule. It's actually a device now it's being commercialized, and the study that I'm presenting are the initial studies about the mostly safety and efficacy of this capsule that were done between 2014 to 2019. So it's under the company Atmo Biosciences now, and the collaborators, the clinical collaborator that I have to acknowledge is Professor Peter Gibson and his colleagues. Okay, so what's the gap? 1 in 5 at some stage of their life suffer from gut disorders, and gastroenterologists, dietitians, they don't have any proper tool. before this capsule for prevention, diagnosis, and monitoring of the gut disorders. Basically, endoscopy and colonoscopy that you heard about it, maybe you have done it. They are very invasive because there are imaging techniques. So the gut should be cleansed, and it's not convenient for patients. Now, question is, can we develop a non-invasive monitoring sensing tool for the gut? Now, in order to do that, we have to identify what the gut is and what we can find in this gut. So we're talking about a 10-meter tubes from basically our mouth that contains a stomach, small intestines that has 3 different sections of the duodenum, jejunum, ileum, and the colon. The materials that we can find and target as biomarkers in this very long tube are microbial communities. Most of them are in the colon, electrolyte, enzymes, metabolites, hormones, and fermentative and chemically produced gases. Which one of them will look? Basically, if the target are the gases, we're talking about hydrogen, methane, carbon dioxide, hydrogen sulfide, and other trace gases. They are the byproduct of microbial and chemical activities which occur in the gut, many of them in the colon. And basically, these gases will release them as fluxes between 0.5 to 1.5 liters every day. And the rest of these produced gases are absorbed through the wall of the gut and goes to the blood circulation, and we exhale them. So an example is like this: carbohydrates, like bread, we eat them. It goes through fermented bacteria. They produce hydrogen and carbon dioxide. If you have sulfur-reducing bacteria, it produces the smelly gas of hydrogen sulfide. If you have methanogens, they produce methane. And also at the same time, they produce volatile organic compounds. First measurements of these gases were done by Professor Levitt back in 1970s. And this is a good example that you see the first paper published. They put tubes in the colon, very invasive, of course, but the outcomes are very interesting. These are non-controlled diet outcomes. As you see, oxygen in the colon is very small, 20.5%. Nitrogen is very comparable to the swallowed nitrogen. Nitrogen doesn't appear in the metabolism of the body, so it doesn't really change by much. Hydrogen is produced at very high concentration. We're talking about 19%, but they're very volatile between one person to another. Some people produce methane. As you see, some people don't. So basically, people from Middle East or Africa, they're methane producers, and the rest are not. And carbon dioxide, 15% on average, is a result of the fermentation. Also, the oxygen drops from the stomach, as you see in the figure, the top figure. The stomach is almost aerobic, full of oxygen. Then it reduces stepwise when we go through the small intestine. And as you see the measurement by Levitt and his colleague, in the colon, the oxygen reduces to less than 0.6.7%. Now, why these gases are important? We are talking about if we can measure these gases correctly, we can recognize inconvenience symptoms. Basically, when we have lots of gases produced, we have belching, flatulence, bloating, and eventually pain. And they are related to malabsorption of carbohydrates or other relevant disorders like IBS. H2, for instance, hydrogen, too much of it has been associated with overgrowth of bacteria in the small intestine or irritable bowel syndrome. Methane is an acidic gas. It causes delayed intestinal motility, basically causes constipation. And too much of it, hydrogen sulfide, is associated with inflammation. If you have IBD, inflammatory bowel disease, or ulcerative colitis, or eventually cancer, the colon produces hydrogen sulfide. NO is a neurotransmitter together with H2S. Now, the story began when about almost 10 years ago. I wrote 7 years, but it's really 10 years ago now. 9 years ago now, that a gastroenterologist came to me and said, okay, we do the bread test analysis. We measure methane and hydrogen bread. If it's more than 10 parts per million, we consider them positive. If it's lower, we consider it negative. But unfortunately, for recognition of, for diagnosis of different diseases of the gut, this 10 ppm fluctuates very badly. The log for correct diagnosis of, for instance, carbohydrate malabsorption is only 70–60%. Can you make it better? So basically, I went back and looked at problems. I said, okay, gases are produced in the colon, the rest of the gut, go through the mucosa of the gut, and then through the cell membranes, and they go to blood. It recirculates, go to the lung, we exhale them. So it's an indirect measurement. Metabolism of the body interfere with them. So this is why, if you read a book, if you walk, all this gas concentration and type changes. The result is like a flip of a coin. It's not reliable. So the guy vanished, unfortunately. I never met him anymore. I never saw him anymore. I don't know what happened to this gastroenterologist, but I became very interested. And I said, okay, we have a solution. How about we put everything, the microcomputer, the sensors, batteries, transmission systems, in a capsule, and we swallow it. And it measures the gases where they're produced, not indirectly, but directly. Okay, so I have still have a good lab of multidisciplinary expertise in medical devices, sensors, functional materials. At the time, I put them together, and my talented group went through developing the sensors and membranes and the rest. None of them were available in the market. So sensors are based on physisorption nanomaterial sensor that I show you. We modulate the heat and become sensitive more or less to different gases. Membranes are based on, for instance, soft polymers that we make them porous, so gases go from one side to another without any hindrance very quickly. So here, for instance, you see these graphene oxides are added, surfaces are not wettable, so they make microcavities. Gases go through the microcavities from two different passive surface diffusion to microcavities, and very quickly from one side to another. If you don't have it, it takes 15 minutes, 20 minutes, half an hour, 1 hour to, regarding different gases to reach equilibrium with inside the capsule. Oops, something happened. Sorry for that. Okay, so this is a schematic inside the capsule. So what is inside is the sensor, which has a heater at the back. And this heater goes from that of room body temperature to 400 degrees. Of course, it's a micro heater, we never feel it. And it's a battery, a silver oxide battery is sufficient to heat it up to that temperature. It heats up and it produces this modulation for the sensor element. At each temperature becomes sensitive more or less to one of these gases. We send it to the microcontroller, we analyze the result, the outcomes. We pre-calibrated them so we can then code the type and concentration of gases, and we send the number to transmitter. One more time, we code it to make sure that many different people that are using different capsules, they can use a capsule at the same time, and we don't mix the results. Goes to antenna, 433 megahertz, come out. So here is a capsule receiver. And what it does, basically every 5 minutes, it sends the information about hydrogen, oxygen, methane, and CO2 out. It has gas permeable capsule. It has microcontrollers that analyze the data. It has transmitter at 433 megahertz, and it's Bluetooth linked that the receiver send this to Bluetooth, and you can see it on your mobile phone. The first tests of the capsules were on ruminants. As you see, fistulated animals, we put the capsules in. This is the result of hydrogen measurements in 3 days. Then we take the capsule out. The result of course, transmitted out at the same time. And we went for a transition of capsule because ruminants, the capsule will stay there. It doesn't go up or down. Pigs, they have more similar gut to human body. We did the animal trials of 56 pigs, and everything went well. The only difference, the only problem is pigs can be quite savage. They can bite you. You needed 3 people to hold them, another person to gavage the capsule inside. So I'm not going to talk about the results, but human being is very easy. You just give them the capsule. You don't need to gavage them inside the body. They just take it, and after 5 minutes, it shows the higher temperature of 35.5, 36.5 degrees, and the measurement starts. So here are the first measurements. As you see, as I mentioned to you, the change of the oxygen concentration is stepwise, and we can see it very nicely: stomach, jejunum, ileum, and the colon. Each step goes, the oxygen drops. So we realize where the capsule is, and we did the ultrasound at the same time to make sure that what we are claiming is correct. Actually, ultrasound showed the transition of capsule from one segment to another. And then hydrogen, as you see, the moment that it passes through the stomach and reaches jejunum, we see a little bit of increase in hydrogen, and then more, and it goes, in this case of example, 3-4% of hydrogen production. That shows that fermentation is happening. Of course, we have lots of CO2 production as well. When we eat, we have a jump in the production of CO2. We have also a temperature sensor as well. As you know, when we walk, body temperature goes up. When we run, it goes even higher. When we sleep, it goes down. And when it leaves the body, actually it goes to room temperature, and we realize that it left the body. Now, the 3 silver oxide batteries inside let this capsule to operate for about 4 days non-stop for measuring the gases, and the temperature sensor for 30 days. So it's quite safe. We exactly know that the capsule stays there, even if it stays there for longer than a few days, the temperature sensor still tells whether it's in or out. We never had any retention anyway. It was very safe so far. Anyway, other experiments is the first person who really did go through all this experiment was myself. I put my body under extreme stress, having a food of very low fiber diet for several days, less than 15 grams. It's not healthy. And very high fiber diet after a week, more than 55 grams a day. Here are the results for low fiber diet. As you remember, you may know, a low fiber diet makes you constipated. It's not something that I advocate and ask anyone to do. It's just a test. So as you see, the second day, the person who was me produced only 0.5% of hydrogen gas. Fermentation was not happening, and eventually that fermentation, even very small fermentation, went to 0. That's actually not nice because it causes gut pain and inconvenience of the gut. And then on the third day, going back to normal food, relief is seen because the hydrogen started to be produced, fermentation started to happen. But again, the pain was there, but we could tell that the day after, of course, when basically capsule and the rest of the materials in the gut are released, this pain should go. So for the first time, we could predict things which are happening to the body. A very high fiber diet, you expect lots of hydrogen to be produced, but it didn't happen. That was very interesting because it was more than 55, 60 grams of fiber per day. It's actually not healthy. What we realized, the fermentation even didn't go up again. And instead, it was quite low. That means I had a very unhealthy diet. And we realized that the oxygen remained very high. That means we were pushing lots of oxygen trapped around the fiber at that kind of concentration of fiber intake. And it was causing the change of the environment of the colon to aerobic. And as a result, the loss of a microbial community happens, loss of 3 kilograms of the body mass. Now, instead of continuing with this extreme diets, we went to, okay, we want to commercialize it, what we should do. Started to play around with the different kind of baselining diet. Here is a high fiber diet and low fiber diet. As you see, high fiber diet, we get different shapes in the responses. But in low fiber diet, we realize, wow, people become the base gas response become very similar from one volunteer to another. As you see, these two are 2 different volunteers. and we could baseline, they became very similar. In this regard, it means if we give them an extra substrate or food above this baseline, then we can see the effect of medicine, food of the person to that diet or medicine. Now, this is the interesting thing. We wanted eventually to compare everything with breath testing. So what we did, we gave people different kind of supplements. We gave them glucose. Glucose gets absorbed by the human body. As you know, it's the smallest sugar. It doesn't get fermented because it has no chance to reach the colon. It gets absorbed through the stomach and the small intestine. Basically, we are talking about 20 and 40 grams of glucose. These are the kind of sugar amount that you find in a can of Coke. And what happened that it gets absorbed through the body very rapidly. This is why we feel energetic after drinking a can. On the other side, if you add emulsifiers like inulin, which is added to, for instance, commercial ice cream or for instance, yogurt to make it fluffy and make it bind together. Those kind of added materials like inulin, as I said, these are longer chain carbohydrates, and they have to reach to the colon to get fermented by the microbial community. Here, we gave 1.25, 2.55, 10, 20, and 40 grams. And when they reach there, they produce loss of hydrogen. So glucose doesn't produce hydrogen, but inulin as a long chain carbohydrate produces hydrogen. Baseline them with low FODMAP diet, which is a very similar to that low fiber diet, but it's healthy, doesn't cause constipation, and it's low in fermentable oligo, monosaccharide polyols and polyols. And we gave these 40 grams of glucose to three different subjects and the same, inulin to 3 different subjects for comparison. Now, these are the magic profiles, magic graphs that you see. So basically, when we gave glucose to people, they didn't produce any hydrogen. Here are the results on the top, are the results of hydrogen measurements of the capsule in different individuals. When we gave them only 1 and 2.5 grams of inulin, the individual produced 1.5 grams, 1.5% of hydrogen, and at 2.5 grams of inulin, they produced 5 grams. As you see, and it increased, went up and up. 5 grams of inulin produced around 8% of hydrogen, and 5 grams of inulin is basically the amount of inulin that you get in a tub, a small 200 grams tub of commercial yogurt. And then after that, we are talking about 10 grams of inulin produced loss of hydrogen. Interestingly, when you look at the bottom graph, and that's a measurement through the breath test analyzer, only for 10 grams, the signal was above, as actually at 10 parts per million. And it shows that the hydrogens produce and we can exhale them. At any other number, any other concentration, it was under the detection limit, reliable detection. And this is the repeatability and association between the breath test and the capsule. So we gave 40 grams of inulin. It's a loss of fermentation happening in these individuals. And we saw that when 23 parts per million of gas could be detected in the exhale of that volunteer, it could produce 8.5% of hydrogen in the gut. And it went up. They were proportional. It was very interesting. 46 parts per million. In the mouth, measurement was proportional to 12.5%, and then 16.5%. The highest that was produced inside the gut was associated with 63 parts per million of hydrogen. Now, the opposite happened as well. It was fantastic. When we gave the volunteers glucose of different concentrations, here is, we couldn't see any gas produced. Here is repeatability for 40 grams, 3 individuals. As you see, is always a 0.5% noise level by measurement of the capsule. But we get one false positive at 10 parts million of hydrogen measurement in the breath of one of the volunteers, which was incorrect. So basically, this is the, as I said, magic of the capsule. It's inconvenient. You take it, you don't need to sit down, you don't need to do the breath test, you don't need to calm yourself down to make sure that there is no noise in the measurements. You walk around, you do whatever you want because it measures exactly what's produced in the colon. It produces 3,000 times more sensitivity than breath test. Signal to noise ratio was more than 20, but for the breath test, less than 2. It was a very interesting product when we published the first outcomes. It received lots and lots of basically interest from media and from people. Eventually, after those, it went through the commercialization, went through human trials of phase 1 and 2. Phase 3 is happening in different countries, including Australia and countries outside Australia. And it actually reached mass production. So every week, tens of these capsules are produced and deployed to different hospitals and different places for the test right now. So I want to finish it by saying that there is a connection to Moderna as where basically Nature Reviews Materials. This paper was published as the major milestones of gastrointestinal devices. And basically, one of the authors, Robert Langer, is one of the major shareholders of Moderna. You heard about Moderna vaccine, everybody heard about it. And they said that the latest, the eventual important milestone for the gastrointestinal devices was basically our capsule. So we're very happy. It's going really well. So basically, I would like to thank you to be present at this session and listening to me. And of course, I have to thank our collaborators and the grant providers and also our colleagues at different partner universities that helped us to do all the clinical trials. Thank you very much.
Thank you very much, Professor Kalantar-Zadeh. Do you have any question?
I'm in my office, but we have ordered that in half an hour we have to leave because of the COVID. So I have to leave my office. And
sorry, but you have a question, please. Just one question from Rocio Canova. He thanked you for your amazing presentation, and he asked for the, yes, you show the capsule advantages. He would like to know if the main drawback of this capsule.
To be honest, it doesn't have any drawbacks because you can take it, you can have your food, and it gives so much information. I tried this capsule already 8-9 times. Anytime I do this, it gives me more information about my body and about my reaction to different foods, about my reaction when I'm sick or when I'm not sick. So it's a wealth of information coming out of this, and we are still feeling amazed anytime we see the signals, new signals in response to new conditions. And as I said, you can take it, you can continue with the day-to-day life, daily life, and there is no disruption.
Okay, thank you very much.
Thank you very much. So I just wanted to apologize that I won't be able to attend the end of the session Q&A because of this. So thank you very much. So goodbye. Have a great day.
Goodbye. Thank you. So now I will introduce the next keynote speaker, who is Dr. Andrei Bratov. He received his PhD in physical chemistry in 1987 from Leningrad State University, now St. Petersburg State University, until 1993, began a senior researcher in the laboratory of chemical sensor of the St. Petersburg University. He was working on the development of ice-fed-based sensor. From 1993 to 1995, he was invited as a visiting professor to the Sensor and Biosensor Group of the Autonomous University of Barcelona. From 1995, He works at the Institute of Microelectronica of Barcelona, currently being a senior researcher in the Biomems group. His main research activities are concentrated on application of microelectronic technology to the development of chemical and biochemical sensors based on potentiometric and impedimetric transducer. Please, Andrei.
Okay. So, okay, thank you for your introduction. I will present you impedimetric sensors, transducers based on 3D interdigitated electrode arrays for bacterial sensor applications. So I'm representing Biomems group from Institute of Microelectronics in Barcelona, belonging to the Spanish Council of Research. Well, if we look for the literature using keywords like bacteria, biosensor, and impedance, we see that during the last 2 decades, the number of publications grew considerably, as well as citations of these articles. So this signifies that the theme, the research theme, is really interesting. So first of all, I would like to remind you what is the electrochemical impedance spectroscopy, because it will be used in all experiments that I will present. If we have 2 metal electrodes in a water solution and apply AC voltage between them, the current starts to flow, and this ratio between voltage and current is by Ohm's law is an impedance, and that characterizes the system, along with the phase shift between the applied voltage and the resulting current. So we can express impedance in several ways, like model of impedance and phase angle, as well as a real part of the impedance and imaginary part. So this graph is called Nyquist plot and presents a real part of the impedance and imaginary part. And it is useful to see clearly what kind of response the system gives us. So if we have a very fast redox couple in the solution that reacts with the metal, then we will have this kind of semicircle used by charge transfer resistance in parallel with double layer capacitance. And this circle is shifted by the solution resistance. If there are some restrictions on arriving of compounds to the electrode, then we'll have a Warburg impedance at low frequencies, and the system will be presented by a classical Randall's equivalent circuit. If there is no redox couple in the solution, then we will have only series system of solution resistance and double layer capacitance. This solution resistance and this straight line present the double layer capacitance. So I have to tell that in all our experiments, we didn't use any external redox couples, so we are working in just water solutions. So to minimize, miniaturize the system, it was proposed to use interdigitated electrode arrays depositing 2 metal comb-like electrodes on an insulating structure and contacting them with the contact pads. The peculiarity of this kind of device is that the penetration of electric field and current into the water solution is equal more or less to the distance between centers of 2 adjacent electrodes. In our work presented here, we work with devices with electrodes 3 micron wide, and the separation between the electrodes is also 3 micron. So this means that the penetration of electric field into the water solution is more or less 6 microns. So, okay, to understand more what kind of equivalent circuit can be used in our devices, we performed the measurements of in the solutions in a very wide range of solution conductivities. And the results are plotted here as an inquest plot or Bode plot, which is a model of theta versus frequency or phase angle. So one would expect that, as in macro case, the measured resistance value is linear in the whole conductivity or solution resistivity range. But it occurs that it's not the case when we are working with interdigitated electrodes. We see that at very low solution conductivities, we see decline from linearity that is produced due to the surface conductivity in the spacing between the electrodes. Just for silicon dioxide surface, it is covered with ionizable OH groups, so that additional counter ions go to the surface to compensate this charge. So in solutions with very low conductivity, the conductivity along the surface may be the same order of magnitude or even higher than in the bulk solution. So this is the reason of this decline. And in reality, this value of RS that we are determining is the parallel combination of bulk solution resistivity, and the surface resistivity of the device. So this principle was used to develop different biosensors, so that we modify with biomolecules that are typically charged the spacing between the electrodes, and register interaction of these molecules with target molecules in the test solution. Once again, as most biomolecules are charged, this reaction provides changes in the surface charge and changes in the surface conductivity. So this is how it looks, that if we'll deposit some biomolecules in the spacing between the electrode digits and measure the current flowing. So we see that part of the current will go along the surface, but once again, taking into consideration that the penetration is around 6 microns, then only small part goes from the surface, and the main part of the signal comes from the bulk of the solution. So to enhance the sensitivity, we have proposed and patented the device that bear barriers between the digits. So the geometry of the sensor is just the same as 3 micron electrode width, 3 micron spacing, but we have a 4 micron high barrier made of silicon dioxide. So what does this mean? This means that the current now goes not through the solution and not from a reduced space, through the reduced space between the digits, but it goes along the barrier surface. So we enhance considerably the reaction of the electric field with the surface of our sensor. So briefly, how this kind of cells are fabricated. So we use a silicon wafer and using standard microelectronic technology, we deposit 2.5 micron silicon dioxide as an insulator on the silicon wafer. Then we use tantalum silicide as a metal layer. By lithography, we form the electrodes. We deposit contact pads, aluminum contact pads, and so the sensor, the flat sensor, is ready. But to form a three-dimensional device, we deposit 4 micron thick silicon dioxide layer using a low pressure chemical vapor deposition method. And then with reactive etching, we open this layer to make contact to electrodes and the contact pads. So this how the sensor looks like. So what this slide present the design of our sensor. So we have a chip 3 by 3 millimeters with electrode with 3 micron spacing. Oh, sorry. Spacing also 3 micron. The barrier height is 4 microns, and the total number of electrodes 216. If we present this like 2 parallel electrodes separated by spacing of 3 micron, the total length of these electrodes will be 16.2 centimeters. So here we see that after etching of 4 micron thick silicon dioxide layer, we obtain nearly vertical walls and vertical barriers. So if we repeat the measurements in water solutions with a wide conductivity range, we will see how the sensitivity in case of 3-dimensional electrode is enhanced. So this is the response of a flat device. This is the response of the device with 2 micron high barrier, and the response of 400 micron barrier sensor is really large. So this permits us to use this sensor as a base for the development of various biosensors. Once again, we are from this graph, you can see that if we fix the conductivity of the bulk solution at a very low level, changes in the measured RS value will be attributed to the surface resistance changes. So while one of the simple modal method to deposit different and manipulate charges on the sensor surface is layer by layer method, and we can use highly charged polyethylenimine as a polycation and polystyrene sulfonate as a polyanion. The method is very simple. We just put in contact the sensor with the polyethylenimine solution, wash the sensor, then we deposit another layer of polyanion, and in this way we can deposit various layers, layer by layer. So let's see how the position of these layers affect the response of our 3-dimensional sensor. So this is the response of a flat sensor without the barrier. This is the expected resistance value, and this is what is measured. In case of polyethylenimine, we see that the charge introduced is very high, so the conductivity of the surface layer is very high. After deposition of polystyrene sulfonate, we compensate this positive charge, and this is the response. While in the range of high conductivities, deposition of this layer doesn't affect the response, and everything is due to the bulk solution conductivity. So once again, if we fix the conductivity of bulk solution at some low level, we can attribute all the changes here to changes of the surface resistance and charges on the surface. So let's go to biosensors. As an example, I will present the use of different sensing elements like polyions, lectins, aptamers, antimicrobial peptides, and bacteria cells. So if we deposit polyethylene imine on the sensor surface, the charge is highly positive. Bacteria are typically negatively charged. So if we put in contact this modified sensor with Escherichia coli and for some time, for 20 minutes, and then measure the spectra and obtain the result of RS in 10 to the minus 5 potassium chloride solution with conductivity around 2 microsiemens per centimeter, we see that even at small concentration of bacteria in the solution, we have the response. The response time is very low. So we see that in 10-20 minutes, we have nearly a biofilm deposited on the center surface. Of course, we can't expect any selectivity from this method. Any positively charged species will be adsorbed on the sensor, but by this experiment, we see that we can see the presence of bacteria on the sensor surface. So another type of active element that may be used is concovalin A biorecognition element. It is an endotoxin that often found on the bacteria cell surface and is maybe dangerous for human cells. For humans, so it is important to determine it. So we tried the concovalin A is selective to lipopolysaccharides, that are toxins, and sorry. We decided to deposit it over the polyethylene imine layer. So here are the changes in spectra. This is the native silicon oxide surface after treatment with polyethylene imine and after attachment of concovalin R to the sensor. So here in this graph, we see the response of RS in time in the presence of 25 micrograms of lipopolysaccharide for sensors modified only with polyethylene imine and sensor modified with polyethylene imine and concovalin A. So we see that there is no real difference between these two responses. So this means that we need to have the selectivity. We need to block the surface covered with polyethylene imine to prevent interaction of lipopolysaccharides with it. So this is the surface modification scheme. So once again, we deposit polyethylene imine, concovalin A, and blocking agents. We use typical blocking agents like bovine serum albumin or Western blocking reagent, but both of these showed poor effectiveness, and so we had to design another method. Another method, was used, taking into consideration very high affinity of glycans to concovalin A. So once again, we deposit polyethylene imine, concovalin A, and then we treat the surface with glycan that is deposited on the concovalin A layer. we repeat this procedure several times, and the optimal design was 2 layers of glycan and concavalin on the sensor surface. Sorry. Here's the response of the sensor with concavalin to the presence of lipopolysaccharides in the solution. So we see that in 10-20 minutes, the reaction is over, and we can obtain stable signal and a very low limit of detection of 2 micrograms per milliliter, which is much lower than reported for other kind of biosensor. The response is a typical Langmuir adsorption curve that in semi-logarithmic scale may be presented as a straight line, and so that can be used for sensor calibration. Another interesting active molecule that can be used for detection of bacteria is antimicrobial peptides. We use synthetic antimicrobial peptide immobilized on our 3-dimensional sensor and studied its sensitivity to Streptococcus sanguineus, which is important colonizer of preimplantitis, the inflammation that leads to the failure of tooth implant in dentistry. Sorry. So in this case, we perform chemical grafting of the sensor using epoxysilane with epoxy group that reacts with the NH2 group of the antimicrobial peptide. In this way, we deposit this molecule on the sensor surface, and we have studied its reaction with the bacteria. Here we see the response of the sensor measured in potassium chloride solution and in artificial saliva. So the linear correlation was found of the cell concentration studied in the ratio of 10 to 10 to 5 colony forming units per milliliter with a sensitivity of 3.5 kiloohm per bacteria concentration decade. And the determination limit was as low as 35 colony forming units per milliliter. So another interesting moiety that can be used instead of antibodies is aptamers that are more stable than antibodies, but have nearly the same and sometimes nearly the same selectivity and sensitivity. So to study this aptamer, we immobilized it on the sensor surface. Unfortunately, typical modification, as in the previous case with epoxysilane, was not good. So we have used thiol modification, thiol modification, and performed the immobilization by the reaction with the disulfide group of the aptamer. In this way, we immobilized this aptamer on the sensor surface. Then we incubated in a sample with bacteria and perform the measurements of the impedance taken as a value, RS value. So we have seen that impedance response increased, well, is linear in the whole range with very good sensitivity. The selectivity of this aptamer in cases when other bacteria strains are present is very high. And what is also important that we can regenerate the sensor surface after the first measurement and perform at least 2 or 3 measurements with the same sensors, which is really important because with the antibody-based sensors, you cannot do this. So to make the regeneration after the reaction with bacteria, we put the sensor for 30 minutes at 80 degrees in water solution. Then to restore the structure, we put it into PBS solution for some time and perform the registration another time. So we see that we totally recover the sensor, the activity of the aptamer after the first measurement. And the last example I want to show is when bacteria used as an active element of the device. So the idea was to deposit bacteria into the well trench between the barriers and study how antibiotics affect bacteria. But as we have seen in the first slides, when we use polyethylenamide, the bacteria is deposited all over the surface, and due to electrostatic repulsion after being on the surface, very little enter between the barriers. So this work was done in collaboration with the University of Groningen and the Medical Center of Groningen. And we used antifouling agent microgels based on isopropyl metacrylamide. So firstly, we modify sensor with polyethylenamide, then we deposit antifouling agent on the PDMS substrate, then by mechanical microcontact printing, we deposit the antifouling agent on top of the barriers, and this prevent adsorption of or attachment of bacteria on top of the sensor, and the bacteria mainly is deposited between the barriers. So this is shows that after microcontact printing, we have the antifouling agent only on the top of the barriers. Then after 20 minutes, we see that most of the live bacteria are between the barriers in the trenches. And this electronic microscopy photo shows that it's really the case. So then we have treated the sensor with ampicillin antibiotic that results in the failure of the cell membrane, and the internal liquid of the bacteria goes into the microcapillary formed by the trench. And so the conductivity when the bacteria is broken goes down due to the higher electrolyte concentration inside. So let's pass to conclusions. We have introduced three-dimensional imperimetric transducers that is highly sensitive compared to its planar analog. And this is due to the presence of insulating barriers, so that the current goes close to the surface of the barrier and gives us a higher signal. Well, chemical modification of the barrier with biomolecules permits to realize label-free detection of some sequence analyzed in the solution due to the surface charge changing provoked by this reaction. So, and the last one, that these kind of devices can be used to develop biosensors of various type for direct label-free detection. Some more information on application of these devices you may find in these publications. And this presentation wouldn't be possible without hard work of the research team, which is highly appreciated. And the research team was formed by Sergei Brosel, Natalia Abramova, Narova Uria, and also special thanks to master science students from various Barcelona universities. Thank you for your attention.
Thank you very much, Andrei. Now, I have just one question.
Yes.
Very interesting, your 3D impedimetric transducers. Are they commercially available?
Well, not in principle. So we make them in our clean room facilities. So if you personally want to make some experiments, I can provide you with these devices, of course.
Okay, thank you very much. Thank you very much, Andrei. Now we will move to the next presentation that will be done by Alice Mieting from the Dresden Technische Universität in Germany. Please, Alice.
So, hello, my name is Alice Mieting, and I'm doing right now my PhD on iron oxide functionalized hydrogens at the Technische Universität in Dresden in Germany. And today I want to present my results on the precipitation of iron oxide and hydrogel resulting in superparamagnetic and stimuli responsive properties of such ferrogels. And what are hydrogels? Hydrogels are a hydrophilic cross-linked polymer which swells under different stimuli. And in my experiments, I use these hydrogel as a template for the wet chemical precipitation of iron oxide by iron salts and then base, resulting in an ferrogel. And what is interesting about these iron oxides is that they have specific adsorption capacities for heavy metal ions like arsenate or something like this. And they also show interesting magnetic properties due to their size. When you have some micrometer size particles, you can see ferrimagnetic properties. And when you have the particles in nanometer size, then you can have superparamagnetic particles, which offer some more physical properties in the end due to inducing heat in the system. So therefore, in my system, I use two different, or I will compare two different types of hydrogel because I want to see the impact of the hydrogel structure due to the formation of the particles. And there I use the hydrogel, which is which is highly cross-linked, and we are using already in sensor systems. And on the other hand, I use an actuator hydrogel. I call it actuator hydrogel because we use it in actuator applications, and this hydrogel has a really low content of cross-linker. It's here called BIS. And this is really elastic and highly stretchable, this hydrogel. And then further, I used the iron salts and the base in the molar ratio to get magnetite. So this means I work with iron 2 and iron 3 iron chloride, which means I need to prevent these iron 2 for the oxygen, and I work in oxygen-free conditions. And furthermore, I have diluted the concentration of these iron salts and the base. So I have an undiluted system, and then I make a dilution of 1 to 10 for each of the solutions, and a 1 to 100 dilution to see the impact of the final amount of the precipitate on the stability for the hydrogel and also on the swelling decrease. So, and what I can see then in a microscopic view, which I have done here in the scanning electron microscope for these undiluted concentrations, I see in the ferrogel a high amount of unresolved particles here in the first column. And then I see some particles which are between 5 to 300 nanometer. And particles in this range, I can see them further in the dilutions 1 to 10 and 1 to 100. And in principle, these particles in the range between 50 and 300 nanometers are fairly well-formed cubic particles. And in the 1 to 10 dilution, I also see some micrometer-sized particles. And when we then do some magnetization curves, I can see for all these samples such S-shaped magnetization curves, which mean there have the whole ferrogels we have analyzed show no coercivity and also have no remanence, so that we can conclude that we have a ferrogel which shows in the compact system a super ferromagnetic behavior. So which need to be further analyzed because there are it's not clear because we have not a really small size distribution in the particles, but the overall view of these ferrogels show this behavior, and we need to do some further characterizations for this. So, and then when I have a look on the swelling properties, I have these gels giving in water, which means there is zero sodium chloride. And then I have put the samples in 1 molar sodium chloride, so which reflects the responsiveness to the ionic exchange. And when I compare these swelling curves of these different ferrogels for the sensor ferrogel and also for the actuator ferrogel, and I compare this with the hydrogel without any particles, I can see a really strong deswelling under ionic exchange, which gives me the sign that there's a really ion-sensitive hydrogel, because in these higher ionic exchange, there will be so some ionic groups will be masked, and therefore there's a deswelling. And when I have a look here at the native hydrogel, you see a little swelling under a higher ionic exchange due to osmotic pressure in such native hydrogel, which has no ionic groups in it. So, and this can be seen for these both types of hydrogels. And what is also visible is with the sensor ferrogel, I can get a relative stable, but maybe on the last cycles not so stable, steady state in the ultra pure water. But when I have a look on the ferrogel, which is more elastic and therefore also more unstable in its mechanical stiffness, I see that there is increasing swelling property with more cycles in water. So this means it's not has not a good stability in water, and the application for sensors is critical and need to be investigated. And now to get a little closer to why there are some ionic sensitive properties in such gels, I will have a look with you at the chemical reaction of these precipitation method. So concerning the time, I will make it a little bit faster. So in principle, when we add iron salts and a base, then the iron hydroxide will precipitate, and then there will be, due to changes in the ionic strength, there will be a formation of these iron oxide hydroxide, and this will give with over the time a solid state reaction, so that there will, I would say, under calcination, such iron oxide or magnetite. So, but I don't have any calcination processes, as you have seen. And so I would say here I have a highly amount of these iron oxide hydroxide, which will reflect these ion sensitive properties of the ferrogel, because this type of iron oxide is really good for deprotonating and protonating. And yeah, this also can be seen when I use the sensor ferrogel over a large range of ionic strength from picomolar to 1 molar, and I will see between the millimolar and the 1 molar a highly decrease of the swelling degree. And for these 1 to 10 ferrogel, here a flattened curve is also seen, which means there's also maybe a sensitive area in a really low concentration area, which in conclusion means that I have, due to this hydrogel, a good template to produce well-formed or well-shaped particles, which show superparamagnetic characteristics, and also have a ion-sensitive deswelling property, which makes these method or the system a promising candidate to use such gels in microfluidics or in medicine, as with these sensitive properties, and also maybe to have also magnetic field guided control and movement. And also I can imagine that it's that there could be a release of thermal energy when you use these gels under alternating magnetic fields, which is also shown in some other publications. And what I will do in my next steps is to use such hydrogels in an piezoresistive sensor and see there the swelling dependency and get a voltage apply for this. So, and in the end, I also want to thank all my colleagues and contributors, in special thank for my colleague Sita Wang for doing the SEM images with me, and my colleague Mia Schlipake for doing the magnetization course. And I want to thank you for your attention.
Now we will move to the presentation done by Rebeca Torrente-Rodriguez, who is from Complutense University in Madrid.
Thank you very much, Nicole. Good morning to everyone. My name is Rebeca Magnolia, and I'm a postdoctoral student from the Electroanalysis and Electrochemical Biosensor Research Group from the Complutense University of Madrid in Spain. And first of all, I would like to thank the organizing committee for giving me this great opportunity to share with all of you some of our most exciting and relevant results obtained in collaboration with the Health Institute Carlos III from Madrid too, regarding the COVID-19 immune response tracking by exploiting the use of novel bioreceptors in connection with electrochemical biosensing tools. By now, unfortunately, all we know the global crisis that COVID-19 has produced worldwide at very different scales, including sociological, economical, but most importantly at the healthcare level. Despite COVID-19 vaccinations finally starting to stem this pandemic tide, however, infectious cases are still increasing, and some regions, especially India and Brazil, continue to be extremely affected by this situation. Therefore, the development of rapid, affordable, and accurate diagnostic tools to try to manage this unique and unprecedented situation are still urgently demanded. As reached through many media outlets, and as you can see from your screens, SARS-CoV-2 virus, the main responsible of the COVID-19 disease development, is composed of 4 main proteins, including the nucleocapsid protein, which is packed in an envelope associated with 3 different structural proteins, namely a spike, envelope, and the membrane protein. It has been demonstrated that of all of them, a spike and nucleocapsid protein are the most antigenic receptors. Therefore, their detection or the detection of specific immunoglobulins against them is crucial for a deeper understanding of how virus actually behaves into our bodies. SARS-CoV-2 virus infection and immune response mechanism are included in a very general manner in these 2 pictures. Briefly, viral particles bind to host cells through the interaction between the spike receptor binding domain and the so-called ACE2 receptor. After several stages, the viral genetic code is finally released into the cytoplasm from our cells. On the other hand, but simultaneously, once the infection is produced, several types of cells get activated and a local immune response is triggered with the subsequent synthesis of different protective immunoglobulins, including IgGs, IgMs, and IgS. In particular cases in which this immune response gets out of control and the production of cytokines is exaggerated, the so-called cytokine storm manifests itself, which can lead to organ failure and even to death. Since SARS-CoV-2 virus is characterized by its constantly changeable nature, therefore the evaluation of immune response nature and longevity by means of the detection of several types of immunoglobulin isotypes once the infection is produced is hugely important for patient stratification, assessment of reinfection, and of course for vaccine development. And due to this variable nature too, as you can see in this slide, there has been established several methodologies for the SARS-CoV-2 virus detection and for the COVID-19 disease management at different stages. Among these methodologies, we can find mainly the so-called molecular and serological based approaches, depending on their use for the detection of very recent or past infection by targeting the viral RNA or the immunoglobulin pattern, respectively. Despite all the above methodologies are of course extremely useful in this regard, however, most of them show important drawbacks and limitations, some of them that I have included at the right part of this slide, and which can be largely solved by the biosensor technology. Simplicity, sensitivity, and selectivity, affordability, multiplexing, and point-of-care capabilities are some of the most remarkable and proven properties offered by this technology. Moreover, some of the most recently reported biosensing tools for the SARS-CoV-2 virus detection and COVID-19 disease management are those based on electrochemical, file effect transistors, and nanophotonic transduction. At this point, it's important to take into consideration the great analytical properties offered by electrochemical biosensors that have been built onto the surface of magnetic microparticles coupled to screen-printed transducers and by using amperometry. These microparticles are widely recognized by their high reproducibility, super-fast kinetics, and almost negligible effect of the sample matrix. At the same time, flexible design of screen-printed transducers fits very nicely with miniaturized and multiplex formats. And regarding the detection, amperometry is widely recognized too, due to be one of the faster, sensitive, and highly selective electroanalytical tools. The integration of all these three, something on which our group is widely recognized, ensures the development of very simple but extremely powerful biosensing devices with great attributes for the analysis and detection of a wide variety of target molecules, of course, including those related with the COVID-19 disease. With all this in mind, our main motivation was the design and development of two magnetic beads-based amperometric biosensors for the nucleocapsid and spike-specific total immunoglobulin interrogation. Briefly, as you can see from both schemes, in-house expressed recombinant spike and commercial nucleocapsid receptors were covalently attached onto different batches of previously activated carboxyl magnetic beads. In a next step, the specific capture of the target immunoglobulins is produced, and then they are enzymatically labeled by using an HRP conjugated secondary antibody cocktail to amperometrically monitor the variation of the cathodic current, which according to our designs is directly proportional to the concentration of the corresponding target analyte by using screen-printed carbon electrodes and in the presence of hydrogen peroxide and hydroquinone as redox mediator. Next, an exhaustive evaluation of each of the key experimental variables affecting both the proper functioning and the response of our proposed methodologies were evaluated by using commercial standards from mouse and rabbit, nucleocapsid, and spike-specific IgGs as target models. As you can see in your screen, we have included a small example of this type of studies, including the concentration of the nucleocapsid antigen directly immobilized on the carboxyl magnetic beads, incubation time of the target IgG, and both the dilution factor and the incubation time of the anti-rabbit secondary, antibody. The optimal values of this layer 3 were chosen based on the highest signal to blank ratio obtained for each of them by comparing the cathodic current in the absence and in the presence of a fixed concentration of each of the target IgG. However, in the case of the concentration of the nucleocapsid antigen, despite we got the highest sensitivity for the highest concentration that we evaluate, finally we choose as low as 5 micrograms per milliliter in order to get a compromise between the sensitivity and the cost per determination. In the next step, and once our biosensors were thoroughly optimized, we demonstrate their suitable performance in buffer solution for the suitable quantification of both types of IgGs, the mouse nucleocapsid IgG, but also the spike rabbit IgG. As you can see from both calibration curves, we did it with great sensitivity, and as you can see also from the low values for the limits of detection that we got for both targets analytes in the few range of nanograms per milliliters, values that are much more lower than the expected concentration for this type of protective immunoglobulins in biofluids from different nature, composition, and complexity, such as serum and saliva. And finally, assuming the great analytical properties offered by our simple biosensing methodologies, we applied both of them for the simple, rapid, and direct analysis or detection of nucleocapsid and spike-specific total immunoglobulins in 1,000 times diluted and previously inactivated human serum samples from control subjects and from PCR-confirmed COVID-19 positive patients. These samples were collected approximately 30 days after the infection onset. As you can see from our data, undoubtedly, it demonstrates the high levels for both the nucleocapsid and spike receptors in the total serological immunoglobulins in the COVID-19 positive samples compared to the control ones, as it can be inferred from the great difference in the amperometric current provided by both types of biosensors in both types of individuals. Importantly, all these results are in great agreement with those provided with one completely different detector reader, such as the Luminex Assay, and therefore verify the extreme reliability that our devices can boast for the accurate diagnosis of accurate monitoring, of the humoral immune response for COVID-19 disease in a sensitive, selective, and very simple manner. And as conclusion, I would like to remark and to emphasize that we have developed and implemented simple, rapid, and sensitive biosensors for the reliable detection of SARS-CoV-2 specific immunoglobulin isotypes against both commercial nucleocapsid and in-house expressed recombinant spike antigens through the combination of magnetic beads and screen-printed carbon electrodes, which are easily transferable on a multi-working electrode platforms for the reliable deciphering of the immune response related to COVID-19 disease in a very simple manner. In fact, all our results show the potential utilities for both tracking the immune response of the COVID-19 positive population, but also for testing the vaccine effectiveness. And of course, I would like to thank all the personal and human resources that have contributed to develop this exciting research project, and to all of you for your kind attention. Thank you very much.
Thank you very much, Rebeca. Now the discussion is open for all the presentation in the morning. Until now, I have no question on the QR box. I have a question for Alice. Yes, Alice. What is the response time of your hydrogel for ionic strength? You show that when you pass from 0 to 1 molar, you have a big increase of volume. But what is the response time?
Yes, the response time is so there I need to do the kinetic measurements, and it's within. I don't have the now up to date the actual graph for this, but it's within minutes and hours.
Okay.
Yeah,
okay.
Depending. So, but actually, I'm working in the free swelling, so I have really large jazz I'm working in, and when I will do this jazz in the sensor system, I will make really less hydrogen. It will be thinner. It will be
it will depend on the volume.
Yeah, and then I expect a faster response time. But now in the free swelling, it will be within hours. So
because you want to use them as stop flow in flow system, so it is difficult. Yes, because you have to have a short response time. Yes.
Yeah, but it's really interesting what will be the exact response time in the sensor setup.
Okay, so can you post your question on the QR box? There is one. Yes, it's good because it is Dr. Motia from Universität of Beknes in Morocco. What type of practical application do you?
The type of particles?
No, in which type of biological fluids you use?
In which type of samples?
Yeah.
Well, we have a lot of experience by using these not only the carboxyl modified particles, but also functionalize with other groups and even proteins and so on. So we have applied from serum to saliva and tissues and RNA extract from cells and from tissues. So they can be, I mean, and the properties and the advantages they have, one of them is that they can be applied to a wide variety of biological X scenarios and with great analytical properties. One of them, for example, in our case, is the almost negligible effect of the matrix. So that's why we can apply almost directly to the fluid we are interested in to perform the analysis.
Rebeca, are you planning to commercialize the test? Can the clinician use the sensor as POC in an easy matter, or do they need training?
Well, this is one of the main advantages, I think, from the electrochemical biosensors. The possible to miniaturize and make it everything portable and very small is one of the main requirements for the point of care testing, right? And of course, we would like to commercialize as soon as possible, but we are at the very beginning of our research. And we have a lot of plans with this methodology, and hopefully very soon we can launch, I don't know, to the market or try to make much more visible to all the people who really need it.
So I think we will close the session, and thank you very much for your presentation, and have a nice day.
Thank you.